Hello everyone. Today we are going to cover compositional similarity and beta diversity, and this lecture will essentially follow the chapter assigned, chapter 6 in Biological Diversity, Frontiers in Measurement and Assessment. So we've talked about alpha diversity and the difference, and we've talked about gamma diversity, overall diversity. And we know that communities differ. And this difference in community composition is termed beta diversity. But how do we compare these communities? Here's two communities in Africa. How do we compare their species composition? We both see zebras, we both see wildebeests. There's giraffes in one and maybe impalas in one and not in the other. So when we compare species composition, we need to be thinking about how similar are the two sites, and we measure that using similarity and dissimilarity indices. So that would be measuring differences between communities, and you can recall that we've already talked about alpha diversity measures, measuring diversity within a community. That would be measuring things like richness, the diversity indices, Shannon-Weiner, and the Simpson's index. We've talked about evenness and relative abundance in a community. So composition comparisons. They're important to both conservationists and ecologists for understanding where species are, how similar certain communities are, and prioritizing for conservationists. And ecologists use it to study homogenizing or diversifying effects of disturbances in both space and time. And to understand how spatial variability contributes to species composition over the landscape. Composition comparisons. Managers. How do managers' actions impact composition when they do treatments such as prescribed fire or grazing at different intensities? And as a practicing ecologist, I've measured both compositional changes to these responses. You can also, managers can use it to understand how human land use affects composition. For example, how do restoration actions affect species composition? And that's the first example I'll show you out of the Cumberland Plain in Australia, in these grassy eucalypt woodlands. So the dots, so what we have here is our typical first two components of the principal components first two components of principal components. And we generate these when we make ordination plots. And while there'll be many axes, most often the most variation is explained by the first 2 to 3 principal components. So here we have these non-intuitive x and y axes that are representing the principal components and the scores that each plot gets for the first and second principal components axis. And here is an example of how managers might use beta diversity to understand how effective their restoration or revegetation has been. So the black dots on the left side are remnant woodland vegetation, and you can see the big ones are from their addition of 33 samples they have, and then an additional 33 samples that they of remnant vegetation, and the small squares are what they collected in this study. And what's interesting is the pasture are the triangles, what they're trying to restore to woodland vegetation, and the triangles are revegetation projects that they decided. So it doesn't appear that they were very successful in their revegetation efforts. If they were successful, the open triangles would be somewhere near the composition of the dark filled-in circles. Here's another example of some work ecologists have done, and this is on the global patterns of bacterial diversity, beta diversity in the seafloor and seawater systems. And you can see, again, we have this typical ordination plot with the first two principal components. Here they used a technique of ordination called NMDS, which is non-metric multi-dimensional scaling. And it's a special form of ordination. We, in this class, tend to use principal coordinates ordination, PCO. And again, they have this separation between pelagic and benthic communities and where the surface vents fall out in seafloor ecosystems. So again, they show this diversity separated by the different types and the different symbols. Here's an example of homogenization of fish fauna across the United States. They used Jaccard's coefficient of similarity here, and they measured similarity through time and measured the changes in similarity. And it's interesting that you can see that zero change in similarity would be the null expectation, but you can see on the right side, everything skewed to the right, that there's been a positive change in similarity. So fish communities across the continuous United States have actually become more similar and caused by homogenization of more interconnected waterways, more introductions from human land use. So basically, the communities are becoming more similar through time. Here's another example of this from the Iberian Peninsula, from France and bordering, or from Spain and bordering France. And these on the left show the similarity within French basins, and on the right, the number of actual species shared. So the dark circles are considering only native species similarity, but when you add in non-native species that occurred, you can see similarity goes up and the slope of the line indicates here the distance to France. So how many degrees is it to France? And you can see that there's a distance decay, and that's something that we expect in island biogeography, that as distance changes, similarity changes. So you have usually referred to as a distance decay effect. So the aim is not just to describe the spatial variation in a community, but understand the causal factors. How do environmental gradients affect the distribution of species? To what contents do compositional differences reflect neutral variation in species abundances due to dispersal limitations? That would be this distance decay that I was talking about. And we're going to evaluate similarity and differentiation. So A, alpha is within, B, beta is between, and the gamma is the whole landscape, just to quickly review. So assemblages. To study composition, we are evaluating assemblages that are a priority, that we a priority consider to be similar. A community is an assemblage of two or more different species occupying the same geographical area. The community area can be defined by a soil type and elevation range, but also by host species. And these differences can range in size from millimeters to millions of hectares, from different layers of bacteria in hot springs to thinking about species composition and shared similarity of shrub steppes around the world, thinking of large scale millions of hectares. So when assemblages are spatially uniform, we can take samples throughout and estimate relative abundance. If they're more heterogeneous, we can estimate mean properties. Identical compositions if the relative abundance of each species is identical, or if we're ignoring relative abundance and the species list is identical, then we'll get identical composition. Maximally dissimilar communities or assemblages if they share absolutely no species. So how similar are these two assemblages? Same species, same relative abundance. But what about these two assemblages? They seem to share no species in common. So they would have a dissimilarity of 1, totally dissimilar. What about these two? This lies somewhere in the middle. We have different relative abundances, but the same species identity. And so this is going to depend much on the metric that we're using to make these measurements. And then again, another example of somewhere in the middle where we have overlapping species in assemblage 1 and in assemblage 2, but then unique species in assemblage 1 and assemblage 2. So similarity is the relative compositional similarity that's going to range from 0 to 1, with 0 sharing no species and 1 sharing all species. And we use this to make inferences about the assemblages based on implicit assumptions about mathematical properties. So similarity, we have two pools. How unique are these? And this is a diagram that I put up during the field week, right? A is the shared species, C is unique to C pool, and B is unique to pool B. So we often talk about terms of dissimilarity, and compositional differences is essentially the opposite, with the metric of 0 being 0% dissimilar and 0 sharing all of the species, so being 0% dissimilar, and 1 sharing no species. So this is the metric. So we use this dissimilarity index, the Bray-Curtis dissimilarity index, in our homework 3 on treatment effects. So there's measures of incidence-based similarity, so presence and absence, and the two most common are the Jaccard's and the Sørensen index, both based on comparing two assemblages. And they differ in the perspective that Jaccard compares the number of shared species to the total number of species, and Sørensen compares the number of shared species to the mean number in a single one of those assemblages, as such the true measure of overlap. So we'll work, here are those two here. So Jaccard's is just A, the number of shared species, over the number of shared species plus those unique to 1 and unique to 2, whereas Sørensen takes the true average, so we have 2 times the shared number of species over 2 times the shared number of species plus the number of unique species in B plus the unique species in C. So here's our example of 2 pools. How many are unique and how many are dissimilar? And here's the example. So how many shared species do we have? 1, 2, 3. What are the richness and the Simpson's diversity of each of those? How similar are those? So if we concentrate on the bottom left, one site had a richness of 7, the west side, and one site had a richness of 6. They both had the same Simpson's diversity indices numbers. And now let's think about those. So those are the within plot alpha diversity values. So now let's think about the beta diversity component of this and calculate the Jaccard and Sørensen's index. So we have 4 species shared in common. 3 species are unique to the west side. 2 species are unique to the east side. So if we use our formulas for Jaccard and Sørensen's index, we can calculate those 2 values. And you can see that the 2 similarity indices come out different from each other. Jaccard shows lower similarity, whereas Sørensen shows slightly higher similarity because of the different weighting of the unique pools. Sørensen's gives greater weight to shared species than those found in only one site. When richness of one assemblage is large and the other is small, for example, one site has 20 species and the other site has 5 species, both indices give low value, which is an accurate reflection, but sometimes it's useful to normalize measures so that we can reach one when overlap is large, as it can be given, as you can have problems when you have two species rich with two assemblages with very large differences in richness. So here's an example of one of these. So we have Lake A and we have Lake B. So one site has 2 species, the other site has 4 species. So even though there are 2 species unique to Lake B, Jaccard and Sørensen would never equal 1. But this value here, by taking the minimum of the 2, would allow it to equal 1. So so if we use this A, the index of A over A plus the minimum, so one of the 2 of A or B, in this case it would be 2, we can arrive at 1. So this is the conditional Sørensen. I think it's also called the beta sim index sometimes. And there are a whole bunch of similarity indices that we can use. And this is only a small example. This is the example from table 6.2 in the chapter you were assigned to read. But there's also a paper by Koloff that you can find in the beta diver function if you want to find it listed in the vegan package. And it lists 24 dissimilarity indices in there. But sometimes we want to make measures of dissimilarity that are more than just based on the presence or absence of a species in an assemblage. We would like to use abundance instead of just incidence. It's ecologically more informative and meaningful. And so in this class, we've tended to use the Bray-Curtis index for those. So 2 sites with the same relative abundance but different absolute abundances of the same species will not be completely similar. And so if we calculate it with the Bray-Curtis dissimilarity, we reach a maximum of about 0.6 here, whereas the Morista-Horn is 0 because the relative abundances are the same. So this Morista-Horn is a good indices to use. And so it's using abundance, not indices. And you can find it in the VegDist package under Method Horn. And it's basically comparing the proportional richness of each species in the assemblage. So when 2 assemblages have equally common species, the Horn and Sørensens all give proportion of the shared species in an assemblage. But Morista-Horn is based on the square distances, so the more dominant species influence more. So if you don't think you detected all the rare species or you have a weird rare tail in your relative abundance, it may be less good. So the Bray-Curtis may be less good for use here. And so it'd be better to use the Morista-Horn because you're making these individual proportional comparisons of the 2. It's an overall good, robust choice. So in summary, diversity and composition. There's a strong connection between diversity and composition. If assemblages are identical, pooling them will leave diversity unchanged. If different diversity increases, comparing assemblages of alpha to pooled to get your gamma diversity provides information on compositional similarity. But using a ratio of alpha to gamma has some issues, but taking the sort of exponential Shannon or the inverse Simpsons helps. For measuring compositional dissimilarity using incidence or presence-absence, Jaccard and the Sørensens are good choices, but are sensitive to differences in richness between samples or assemblages. For abundance-based similarity measures, the Morista-Horn or the Bray-Curtis work well, but Bray-Curtis has an abundance bias when one assemblage has much higher levels of abundance than the other assemblage. An estimation of similarity and differentiation is statistically challenging. You can see that more in the book. And we have been in class using this permutational analysis of variance. So basically making regression models of dissimilarity based on Euclidean distance dissimilarity of our predictor variables, but doing it in a permutation way by removing and taking out variables till you calculate conditional R squares. And that's done in the Adonis function in the vegan library. So I hope you guys enjoyed it. I hope you guys enjoy the class. Thanks for listening.